Welcome to Planetarian Daily. I'm Patrick in Vancouver, and this is episode 43. Every week we sit down with the newest peer-reviewed papers that are quietly reshaping how we understand life on Earth, from the bottom of the ocean to the edges of the cosmos, and from the vineyards of the American Southeast to the inner workings of our own cells. Today's stories move across vastly different scales, yet they all circle around the same theme. Systems we once thought were fixed or simple are turning out to be far more dynamic, regulated, and recoverable than we assumed. Let's begin with something that could eventually land on your dinner plate. Researchers working on muscadine grapes have identified reliable genetic markers that can tell whether a young vine will produce seedless, self-pollinating fruit years before the plant ever flowers or sets its first bunch. Muscadines are a distinctly North American species, prized for their thick skins, intense flavor, and natural disease resistance, but traditional breeding has been painfully slow. Vines take 3 to 5 years to fruit, so every generation of selection tied up land, water, labor, and patience. Now, breeders can test tiny seedlings in the greenhouse, identify the ones carrying the desirable combination of seedlessness and self-fertility, and cull the rest immediately. That compresses what used to be a decade-long cycle into something far more manageable. My take is that this is a perfect example of precision breeding done right. It doesn't require controversial gene editing. It simply accelerates natural variation that already exists within the species. The same marker-assisted strategy has already transformed tomato, maize, and wheat breeding. Seeing it applied to a specialty fruit like muscadine suggests the toolkit is maturing and can reach smaller but economically valuable crops. It's a quiet win for agricultural efficiency and for anyone who loves a good glass of muscadine wine or a bowl of fresh grapes without spitting seeds. While we're learning to read the genetic future of individual plants, a very different kind of long-term forecast is coming from the Potsdam Institute for Climate Impact Research. Their latest modeling explores what would happen if the Atlantic Meridional Overturning Circulation, the massive conveyor belt that moves warm surface water north and returns cold deep water south, were to shut down or seriously weaken. The surprising finding is that a collapsed AMOC wouldn't just cool Europe. Over centuries, it would also reorganize circulation patterns in the Southern Ocean. That reorganization, the models suggest, would allow the Southern Ocean to release a portion of the carbon it has been storing for hundreds of years. The extra carbon dioxide entering the atmosphere would add roughly 0.2 degrees Celsius to global temperatures, even if all other climate forcings remained stable. The study is a reminder that the ocean isn't a passive reservoir. It's an active, slow-moving participant in the carbon cycle. These processes unfold on timescales that dwarf political cycles and even human lifetimes, which makes them easy to ignore in day-to-day policy debates. Yet the work underscores that some of the climate system's most important feedbacks may still be ahead of us, not behind us. It's sobering, but it's also the kind of insight that helps us ask better questions about where we place marine protected areas and how we monitor deep ocean carbon stores. Fortunately, not every ecological story this week is about slow-motion disruption. In Ecuador, researchers have been tracking how tropical rainforests regenerate after being cleared for agriculture. Their data show that both tree species diversity and vertebrate species diversity can return to more than 90% of primary forest levels within about 30 years if the land is simply left alone. That's dramatically faster than many conservation biologists had assumed based on older chronosequence studies. The team followed multiple sites that had been pasture or cropland and then abandoned, meticulously cataloging everything from canopy trees to understory birds, bats, frogs, and small mammals. What they found was a remarkably orderly return. Pioneer species give way to mid-successional trees, which in turn create the microclimates and food resources that allow shade-tolerant primary forest species to reestablish. Animal communities follow the vegetation, not the other way around. The speed of recovery offers measured but genuine hope for natural regeneration strategies across the tropics. It doesn't mean we can stop protecting intact primary forest. Old-growth ecosystems still contain unique microhabitats and enormous carbon stores. But it does suggest that with thoughtful land use planning, we can allow working landscapes and recovering forests to coexist more successfully than we once feared. For countries trying to balance rural development with biodiversity commitments, this is encouraging news. That theme of tightly regulated biological recovery brings us to one of the more fundamental discoveries in stem cell biology this year. Researchers have formalized the existence of what they're calling a stemness checkpoint, a molecular decision gate that actively polices whether a cell maintains its stem-like properties or commits to differentiation. For a long time, the popular image of a stem cell was a completely blank slate, passively waiting for signals. The reality, it turns out, is far more active and precarious. Inside your bone marrow, your skin, your intestinal lining, and many other tissues, this checkpoint constantly integrates signals from neighboring cells, nutrient availability, and mechanical forces. It builds on a key 2008 insight that true self-renewal often requires the active suppression of differentiation pathways rather than simply the absence of differentiation signals. The new work demonstrates that this same logic operates across many different stem cell populations and across developmental stages from embryo to adult. The practical implications are significant. Small molecular shifts at this checkpoint can push a population from normal tissue maintenance into accelerated repair, or, if the mechanism is broken, into the uncontrolled proliferation we see in cancer stem cells. That means future therapies could target the checkpoint directly, either to enhance regeneration in degenerative diseases or to force malignant cells to grow up and stop dividing. I find this story quietly profound. It reframes regeneration not as an open-ended possibility, but as a carefully policed boundary. Understanding that boundary better will likely shape both regenerative medicine and oncology for the next decade or more. Keep an eye on this one. It's foundational rather than flashy, and foundational work tends to have long legs. From the molecular machinery that controls what cells are allowed to remain immortal to the very practical tools that help breeders create better fruit. This week's research shows science operating at both ends of the telescope. Those muscadine grape markers I mentioned earlier aren't just a laboratory curiosity. They could shorten the breeding timeline from 12 years to roughly 3 or 4. That means new disease-resistant, flavorful, seedless varieties can reach farmers and consumers within a realistic planning horizon instead of outliving the original breeder. The same approach is already being piloted in blueberries, kiwi fruit, and certain citrus lines. It's a clear demonstration that targeted non-GMO biotechnology can reduce land, water, and chemical inputs while improving the sensory quality of what ends up on our plates. In an era when consumers want both sustainability and flavor, this feels like genuinely aligned progress. Yet even as we improve managed systems on land, the ocean continues to reveal the sheer scale of our industrial footprint. The first truly global inventory of bottom trawling catch has now been compiled, and the numbers are sobering. Trawlers are catching more than 3,000 fish species, and the true total could be nearly twice that when underreported bycatch is included. Many of those species are already flagged as threatened or near threatened on the IUCN Red List. For the first time, we can see the planetary footprint of this single fishing method laid out in cold data. The study doesn't call for an outright ban. Bottom trawling supplies a meaningful portion of global seafood, but it does provide the quantitative baseline that policymakers have lacked. It should sharpen discussions about marine protected areas, gear modifications, and the trade-offs between cheap fish and intact deep-sea ecosystems. The same impulse to question long-accepted industrial practices appears in livestock research this week. A carefully controlled study on pig farrowing systems compared traditional restrictive crates with larger, more open pens that still meet biosecurity standards. The headline result is that piglet mortality rates were statistically identical between the two systems, while sow health, locomotion, and stress indicators were markedly better in the open housing. For decades, the industry has assumed that tight confinement was necessary to prevent crushing and to maximize output. This work suggests that assumption may have been overly pessimistic. Improving welfare without sacrificing productivity is one of those rare, genuine win-win opportunities in animal agriculture. The findings are likely to fuel renewed debate over housing standards in Europe, North America, and wherever pork production is scaling up. They also remind us that welfare science has moved beyond simple sentiment. It now delivers hard production data that can inform regulation and consumer choices alike. Shifting from the bodies of pigs to the bodies of people, an analysis of healthcare spending across U.S. metropolitan areas turned up a striking absence of pattern. When researchers ranked the 10 highest and 10 lowest spending regions, they could find no consistent demographic, economic, or structural explanation for the differences. Some high-spending areas had older populations, others had younger ones. Some were rich, some were poor, some had lots of specialists, others relied more on primary care. The variation, the authors conclude, looks less like rational adaptation to local needs and more like evidence of deep systemic inefficiency. This isn't a new observation, but the geographic lens adds weight. It suggests that enormous amounts of money are being spent in ways that don't reliably track health outcomes, local disease burden, or even price levels. In a time of strained public budgets and rising insurance costs, these data add fuel to calls for more evidence-based, regionally tailored approaches to resource allocation. The study won't solve the political problems of American healthcare, but it does make the irrationality harder to ignore. On a more hopeful note for everyday well-being, 2 studies this week reinforce how much leverage we have over our own mental and physical health with relatively simple tools. The first establishes a clear statistical link between a strong sense of meaning or purpose in life and lower rates of depression, even after adjusting for many other known risk factors. The finding sits comfortably alongside decades of research on social connection, exercise, and sleep. It suggests that purpose isn't just a philosophical luxury, it functions as a measurable psychological nutrient. For anyone listening who struggles with low mood, the message isn't just find meaning and you'll be cured, but rather that deliberately cultivating purpose through relationships, creative work, volunteering, or spiritual practice may be one more evidence-based lever worth pulling alongside therapy or medication. Closely related, a randomized controlled trial using wearable sleep trackers tested a digital mindfulness program delivered via smartphone. The intervention was modest, no week-long silent retreats, just short daily practices, and yet participants showed objectively measured improvements in sleep efficiency, fewer nighttime awakenings, and healthier heart rate variability, a marker of autonomic nervous system balance. What I like about this study is that it moves mindfulness out of the realm of self-report questionnaires and into hard physiology captured by consumer devices. That credibility matters. It suggests that scalable, low-cost digital tools can produce genuine biological change without requiring huge time commitments or expert instruction. In a world where good sleep remains elusive for so many, this is the kind of pragmatic finding that deserves to be shared. Staying with the developing brain, new longitudinal data reveal how cognitive profiles diverge between adolescent boys and girls. The researchers tracked mechanical reasoning, academic skill acquisition, and processing speed across the teenage years and found that the trajectories are not static, they continue to shift throughout adolescence. Underlying much of the difference appears to be the pace of maturation in basic mental processing speed, which follows somewhat distinct timetables in males and females. This isn't about declaring one sex better at anything. It's about mapping the normal, healthy variation that exists during a critical window of brain development. The hope is that such maps will eventually help educators, counselors, and clinicians tailor support more precisely, whether that's adjusting teaching methods, recognizing when a student's struggles are developmental rather than permanent, or simply offering reassurance that the awkward, uneven progress of the teenage brain is proceeding on schedule. Adolescence is messy by design. The better we understand its choreography, the more compassionately we can guide young people through it. Finally, 2 stories that bookend the episode, one from the very large and one from the very small. Cosmologists have put forward a new framework suggesting that dark matter may exist in 2 distinct phases or states. The proposal, published in the Journal of Cosmology and Astroparticle Physics, offers a potential explanation for why detectors have seen null results in some energy ranges while still leaving room for dark matter to be abundant elsewhere. It adds a layer of complexity to the search, but also a new set of testable predictions. Like many ideas in fundamental physics, it may turn out to be wrong, but it illustrates how seriously the community is treating the continuing absence of a direct detection signal. And on the longevity front, the latest results from the National Institute on Aging's Interventions Testing Program delivered a string of negative findings. 8 compounds, astaxanthin, meclizine, metaglitazone, pioglitazone, alpha-ketoglutarate, mifepristone, methotrexate, and a combination of atorvastatin plus telmisartan, failed to extend lifespan in the genetically diverse UMHET3 mouse model. In science, negative results are often underappreciated, yet they are crucial. They prune the enormous tree of possible interventions so that time, money, and animal lives aren't wasted chasing pathways that don't deliver. The program continues to set a high evidentiary bar, and that discipline matters if we want to separate genuine longevity compounds from the hype. Before we wrap up, I'll just echo my earlier point. The stemness checkpoint story feels like one to watch. The way cells decide whether to stay young or grow up is so central to both regeneration and cancer that any new molecular handle on that decision could ripple through medicine for years. That's the science and health news for this week. If one of these stories sparked your curiosity, share it with someone who likes to think about how the world actually works. I'm Patrick in Vancouver. Thanks for listening. We'll talk again soon. This podcast is curated by Patrick, but generated using AI voice synthesis of my voice using ElevenLabs. The primary reason to do this is I unfortunately don't have the time to be consistent with generating all the content and wanted to focus on creating consistent and regular episodes for all the themes that I enjoy, and I hope others do as well.